couple of minutes. Should have some people soon. Hopefully. Otherwise, I'm going to be talking to an empty screen or an empty group of people. Makes me feel nervous. Okay. Guess if people join, they join. If they don't, they don't. Alright. Guess I'll go over what we're doing this week. Alright. So first thing, of course, there's week 10 notes. I've got week 10 materials, which has our subject matter, all our lesson materials located here. Obviously, I'm going to be putting this recording up as well. And Punnett Square practice. I'm going to walk through a couple of these with you. Once we finish up here, we're going to do a regular Punnett Square, just a monhybrid and a dihybrid. So, without further ado, I guess, and I'm going to leave the music going because y'all know that quiet kind of freaks me out, so it's kind of my way of, I don't know, cutting back on the anxiety, I guess. So, alright, well, if people join, people join. If they don't, they don't. Alright, you're not going to need any of that because obviously we do not have that type of material on virtual. So, that's all. Alright, we're going to go into Chapter 12, Sexual Reproduction and Genetics. We're also going to be discussing DNA in this section. Last week we looked at the cell cycle and mitosis. We're going to start off with Mendelian genetics. Where does an organism get its characteristics? How are different forms of a gene distributed? So the three words that we're going to focus on right now are fertilization, trait, and segregation. Now, fertilization, you could think of that just the same way as you use fertilizer. That lets things grow a lot better. And fertilization is the first section that we're going to talk about in meiosis. Traits and segregation we will get to later. So the first thing is that we'll have a short video covering Mendel's pea plants.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers his lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Okay. Oh, darn it, it went out of the screen again. All right. All right. Do I still not have any participants? No, I don't. All right, so that's going to bring us to the first vocab word, which is genetics, just the science of heredity, how we see how things move from one form down into the next. We can do this through a process of, my brain is not wanting to work for me today. Pedigrees, that's the word I'm looking for. So for instance, as you can see right here, we can tell that these are the two parents and their offspring. So we ended up with all yellow. However, if we allowed one of them to self-reproduce, which means that it reproduced with itself instead of having a secondary parent, then we had offspring that had this ratio. This is a 1-to-4 or 1-to-3 ratio. Again, we allowed each of these to self-reproduce, and if we took all of the offspring, we added it all together, it was still a 3-to-1 ratio. So this was Mendel's experiment and how we came up with the recessive and dominant traits, what he coined. That's going to bring us to the secondary vocab word, which is fertilization. This is what happens in meiosis and sexually reproducing organisms, such as most species of animal, and we'll get into the exceptions of that later. But for this specific lesson, fertilization, we're just going to talk about male and female reproductive cells. In this case, we have both sperm and egg cells that create a new organism. And traits are just the characteristics that you pass on. For instance, eye color, skin tone, hair color, which we will get into later. So that's going to tell us about Mendel's experiment, how he grew pea plants that produced only yellow or green peas. Typo. They self-reproduced. And what he did was he took one that was green, one that was yellow, and combined them to make something called a hybrid. And a hybrid are offspring of crosses with parents that have different contrasting genetics. So in this instance, we have a panther and a leopard, which is a hybrid. And then we have a zebra and donkey, which has the beautiful, beautiful name of Zonkey. These are just things that have two totally different characteristics that contrast with one another. For instance, the donkeys, usually brown and gray fur, coated with the, coupled with the zebra's stripes. So on Mendel's experiment, after he created these hybrids, which were the offspring, he cross-pollinated the green and yellow plants. After his first filial generation, that is these right here, he allowed the offspring to self-fertilize. So they reproduced with themselves. So from this, he ended up with a perfect 3-to-1 ratio of their characteristics, which was green and yellow. And that brought us to recessive and dominant traits. He concluded that the two forms of the seed trait were yellow and green, which meant that there were two separate forms. That is what an allele is. An allele is just the alternate form of a gene. So for instance, brown hair and red hair, I'm speaking from experience, obviously, those are two different types of allele for hair color. And from that, we develop the principle of dominance. Some alleles are dominant, others are recessive. For instance, blonde hair is a recessive trait, blue eyes is a recessive trait. We'll get into red hair a little later because it's a little bit different from just being dominant or recessive. But some of them are going to be dominant. So for instance, in the example that we have on the screen, that one cat is dominant, it's taking over the space and causing the other one to recede. So dominant traits are things that dominate the gene, which means they're going to show up no matter if they're there or not. So in this instance right here, we have one dominant gene and one recessive, but we still end up with yellow peas, and that's because yellow was considered to be the dominant gene. However, recessive also are able to be produced if the offspring inherits both of the recessive traits. So during meiosis or fertilization, you inherit one set of traits from your mother, one set from your father. During that fertilization, each of the chromosomes line up, and these become the blueprints of what you are. If you inherit both recessive traits from both mom and dad, you end up with that recessive trait. So for instance, my mom and my dad, since I'm purely redheaded, have the recessive trait for red hair. When they reproduced and unfortunately got stuck with me, my hair was all red because I inherited both recessive traits. And we're not going to talk about segregation too much because it doesn't apply a whole lot just yet. We will, we'll discuss that at a later time. It's going to bring us to the vocab words of what we're going to move into meiosis, which is gamete. It's a sex cell with half the chromosomes, and N is used to show the number of chromosomes in one single gamete. So for instance, on fertilization, they're going to have double. No questions. Obviously, there's still no viewers. So if you have a question, hopefully you email it to me before 3:30. All right. It's going to bring us to Mendelian genetics, how we're going to use probability to predict certain traits, how alleles segregate when more than one gene is involved, and what did Mendel contribute. You're not going to need that. All right, application of genetics. This is more so where we're going to see a lot more math involved, but it's real simple math. I know you've probably seen a Punnett square before, hopefully. God help me if you haven't. God, it feels weird teaching to a screen. I may end up like getting cardboard and putting people's faces on it or something like what was it? Some team in Denver did that. I'm just talking to myself. Anyway, this is an introduction to phenotypes and genotypes. And we're going to start off by the two words homozygous and heterozygous. Obviously, those of y'all that have me for physical science, you know the difference in the roots or the prefix homo and hetero. Homo meaning same, hetero meaning different. So if something is homozygotic, that means that it has two of the same. So they are the exact same alleles. Something is heterozygotic, that means they have two opposites or two different traits. If you are heterozygonic dominant, then no matter what, the dominant trait is going to show up. However, if you are homozygous recessive, then the recessive trait shows up. So the only way for that recessive trait to purely show up is homozygous recessive. Okay, that's going to bring us to genotypes and phenotypes. Genotypes refer to the genes in particular, whereas phenotypes are things that you can see. You know, photons are light energy or light particles, and you can think of photos. Phenotypes are things that you can observe, you can see. But genotypes are things that we have to look directly at the DNA in order for us to determine. So, Mendel allowed the large Y, little y plants to self-fertilize. That's referring to the dominant and recessive. When you see a capital letter in terms of genetics, it's going to be dominant. Recessive ones are going to be lowercase. Yes, that's big. So a cross involving two hybrids for a single trait is called a monohybrid cross. Once he saw the patterns, he began to look for other traits, also the wrinkled peas. Instead of being just yellow or green, he looked at wrinkled or smooth. And that gave us a dihybrid cross. This is something that shows the probability of two traits and are heterozygous for both, which means that they inherit both the dominant and recessive traits. So it's going to be a cross between both of them. And law of independent assortment, all that means is that there's random distribution of alleles during gamete formation. And that's going to bring us to Punnett squares. Now, I'm going to work with you on a Punnett square here in just a minute. I think it's in the next slide, yeah. Punnett squares are used to predict the results in genetics. So we're going to do a simple monohybrid cross. Let's see, draw. Yeah, let's draw. All right, so let's say that we had, I want it to be, it's going to need to be a brighter color. Let's go with red. All right, so let's say, I'm trying to see, I think, let me see, hairy and hairless guinea pigs. Okay. So let's say that we reproduce two heterozygotic guinea pigs for their hair. So that means that we have, since they're heterozygotic, I cannot draw with a mouse. It means that they're going to inherit both the dominant and the recessive. If we want to see the probability of their offspring inheriting the hairless genetics or the hairless phenotypes, all we do is we plug it in. So we take the mom, which I normally put on the top. It doesn't really matter where you put it, but you take the top and the side and put them together. So this one would be homozygous dominant. Okay, again, this is H, so since it's recessive, it's going to come second. Always put the dominant one first. Makes things easier. This is heterozygous dominant. Okay, now let's come over here. Big H, little h. Okay, this is heterozygous. Ooh, my brain. Heterozygous dominant. Yeah. Sorry, guys. It's early. And finally, that brings us to the little h again. Took the top and the side, little h. This is homozygous recessive, which means that out of all of this, we have a 25% chance, because 25 goes into 104 times. How do I clear it? Oh no. Oh no. Annotate, clear. There it is. Clear all drawings. Awesome. Great. All right, moving on. Oh no, what have I done? There we go. All right. Hybrids have a trait from each parent. We already talked about that. Homozygous have two alleles that are the same. Hetero have two alleles that are different. We are not doing balloon babies. I am sorry. Anyway, it's going to bring us to the work for today. So the first one, you'll see that there are two separate things attached here. Make sure to complete both sheets as each one is worth 50 points apiece, bringing the total assignment grade to 100. I will be doing the first few problems of each sheet with you in either the Zoom meeting or the video. Obviously, this is going to be video because, again, nobody's watching. So the first one, we're going to come into genetics practice horses. Each of you got your own unique worksheet. So if it can manage to not poop out on me. All right. So you're going to read this section. Horses' gaits are influenced by whether the animal inherited one, two, or no mutated copies of DMRT3, the so-called gatekeeper gene. Horses without the mutation generally show the regular gaits of walk, trot, and canter. Heterozygous horses have received the normal gene from one parent, my apologies, and the mutation from the other parent. These horses can still produce regular gaits. Homozygous horses have received mutated genes from both parents. These gated horses can usually walk and canter and may be able to trot, but most are likely to pace. The pace is a lateral two-beat gait. In the pace, the two legs on the same side of the horse move forward together, unlike the trot, where two legs diagonally opposite from each other move forward together. In both the pace and the trot, 2 feet are always off the ground. So that's where that's our reading assignment. The flaxen gene is a trait that causes the mane and tail of chestnut-colored horses to be noticeably lighter than the body coat color, often a golden blonde shade. It is thought to be recessive, but may be controlled by multiple genes. All right, so this one right here, it's going to ask you what are the genotypes and phenotypes possible in horses. So, in order for us to do this, let's see, is this? Yeah. We're going to insert table. This is going to be genotypes and phenotypes. So, insert table. This is going to be a 4x4, 3, 4. All right. So we have, let's see, we have a big T, little t, big F, little f. Let's scooch this one over. Guess I need to insert another column to the left. All right, now I can. So all right, got big T, little t, big F, little f. So we can end up here. This would be T. T, F. Oh, Jesus. Come on now. Alright, I'm going to have to post this in the new one. I've got to redo this one. This document's not wanting to work for me, but I will post it. So, give me one sec.